Assalamualaikum and hi everyone. Today we are going to present about the determination of flower pigments using paper chromatography. Before we start, let me introduce to my group members, which is me, Farah, Amira and Humaira. Without further ado, let's start with this presentation.
Hi, my name is Amira Auni. So I'm going to share with you guys what is definition of chromatography. Thus, chromatography is a potent analytical method that enables researchers to split complicated mixtures into their constituent parts according to variation in solubility and how the stationary phase, which is the paper, and the mobile phase, the solvent, interact. Also, the importance including understanding plant biology. For example, flower pigments such as anthocyanins, carotenoid, and the chlorophylls play vital roles in plant processes. Overall, chromatography is widely used in laboratories for analyzing substances in fields like chemistry, biology, and environmental science. Next, the technique used in this experiment is paper chromatography. So what is paper chromatography? It is an analytical technique used to separate particles on the basis of their polarity towards the mobile phase and stationary phase. Also, the paper chromatography is a type of solid-liquid partition method, which is the stationary phase is the cellulose filter paper and the mobile phase is liquid. Then why use this technique? This technique improves the time efficiency through the process. It can be completed within a few hours from sample preparation to the observation of results and make it suitable for educational purpose and research applications. As well as low cost, the material required for paper chromatography such as chromatography paper and common solvents like acetone or ethanol are inexpensive and readily available. The great thing is the setup is also relatively fast and does not require any expensive equipment. Moving to the next part, which is the pigment that can be found in flower. In general, 3 pigments can be found in flower, including yellow or orange, red or pink, and green. For yellow or orange pigment, carotenoids are another significant group of pigment that provide yellow to orange colors in flower. These pigments are essential for photosynthesis and play a role in protecting plants from photoimage. Carotenoid can coexist with anthocyanins in flower, contributing to a broader spectrum of colors. Other than that, red or pink pigment typically associated with anthocyanins, which can vary in shade based on pH. Anthocyanins are water-soluble pigments belonging to the flavonoid group. They are primarily responsible for red, purple, and blue colors in flower and fruits. The main type of anthocyanidins include cyanidins, delphinidins, pelargonidin, peonidins, pitonidin, and malvidin. Also, the green pigment from chlorophylls, which may appear as a background color if it presents. The chlorophyll are the green pigment found in chloroplasts. They absorb light primarily in the blue and red wavelength while reflecting the green light. For the flower sample used in this study, Nerium indicum known as the oleander, it has ornamental use, which is oleander cultivated for its beautiful and fragrant flowers, which can be white, pink, or red and bloom from late spring to late summer. This flower can be found in native to the Mediterranean region. Then, the rose centifolia, known as the cabbage rose, Provence rose, and rose de May, or known as the flower of May. The flower is characterized by its many petals, often exceeding 100, giving it a lush appearance. This rose thrives in temperate climates and is commonly found in regions like France. The last sample is Zara coccinea, it known as jungle geranium and burning love that reflect its vibrant flower. Ixora coccinea produces vibrant cluster of tube-shaped flowers in color such as red, orange, yellow, pink, and white, blooming year-round under ideal conditions. It can be found in tropical Asia, including India and Sri Lanka.
The objective of this experiment is to extract flower pigments from 3 different types of flowers and separate them by using paper chromatography. The second one is to identify components of flower pigments from the 3 different types of flowers by calculating the RF value. Paper chromatography is considered to be the simplest and most widely used of the chromatographic techniques because of its applicability to isolation, identification, and quantitative determination of organic and inorganic compounds. There are 2 phases in paper chromatography, which is stationary phase and mobile phase. The stationary phase is the moisture or water present in the pores of cellulose fibers that is present in the filter paper. The mobile phase is the solvent used. The type of separation here is partition chromatography because the substances are partitioned or distributed between the liquid phases. So how does paper chromatography works? The chromatography paper is made of cellulose, which is a polar compound. As the solvent, which is the mobile phase, travels up the paper, it carries the component of the mixture with it and the separation of mixture takes place. The non-polar compound will travel farther with the solvent because they are weakly absorbed by the stationary phase. The more polar compound will travel at a slower rate because it has a strong interaction with the stationary phase, which is a polar compound.
Hi, I'm Humaira. For the methodology part, I'm going to explain to you one by one about the step preparations needed to find the flower pigments using a chromatography paper. First, a fine parallel line in about 2 to 3 cm across the strip using fine pencil is drawn straight from one end of chromatography paper edges. Second, the flower petals are gathered to grind and some more ethanol or flowers petals are added until a very dark concentrated liquid are extracted. Third, using a capillary tube, the liquid extract was transferred into the chromatography paper. Literally at the center part of the parallel line was drawn. 4 until 6 loads of extract could be done. However, before adding the next load, the paper must be fully dried first. Let it be 2 to 10 minutes. Fifth step, the paper strip was placed into the solvent container. Make sure the level of the solvent touched the paper strip, which is the mobile phase, was below the extract line. Sixth, the chromatogram then was removed after the solvent front reached 2 to 3 cm from the top of the paper. For the next step, a pencil used to mark the location of the solvent front, and then let it be for a few minutes. And that's how step 1 to 7 was repeated for 2 other different flower specimens.
These are an example of result for 3 different flower species, which is Nereum indicum, Rosa centifolia, and Izora coccinea. The RF value of each of the flower was calculated using the formula of RF equals to distance traveled by the component divided by distance traveled by the solvent. The RF value of Nereum indicum is 0.62 and the RF value for both Rosa centifolia and Izora coccinea is 0.58. The result was compared to the standard RF values. The standard RF values of anthocyanin ranges from 0.32 to 0.62. Therefore, it is confirmed that all flowers have the presence of anthocyanin that is responsible for the color of red and pink. So what makes the RF value different from each flower? It has to do with their polarity. Although all of the flowers have the same pigment, which is anthocyanin, but they may have different structure of anthocyanin that affects their polarity. Nereum indicum have RF value of 0.62, which indicates that it travels the farthest and have low polarity because it is weakly interact with the stationary phase. For both Rosa centifolia and Izora coccinea have the RF value of 0.58. These indicate that they are more polar compared to Nereum indicum because they have a little less distance travel, which makes them strongly interact with the stationary phase.
In conclusion, the experiment successfully achieved its objective. The paper chromatographic method was indeed proved to be an efficient and reliable technique for extracting, separating, and identifying flower pigments. Moreover, by isolating the flower pigments and comparing the results with their RF standard values, it is confirmed that the presence of anthocyanin that was identified in all flower specimens are responsible for the coloration observed in the flowers. Lastly, this experiment also provided valuable insights into the diversity of pigments present in different flowers.
And that is all for our presentation. Thank you for listening.